welcome everybody, and I'm very excited today to introduce our very own GGI department faculty member, Dr. Athena Starlard-Davenport. So Dr. Starlard-Davenport began her scientific career at the University of Arkansas for medical sciences, where she received her PhD before being selected to participate in the FDA Commissioner's Fellowship Program, where she worked on epigenetics and breast cancer. Her passion for research into the genetic mechanisms underlying breast cancer health disparities led her back to the University of Arkansas to pursue a second postdoc. In 2016, she joined the faculty of the GGI department, fortunately for us, and she has continued her research on the molecular cancer epidemiology, risk assessment, and breast cancer disparities, and also started new and exciting research on epigenetics of globin gene regulation and sickle cell disease and the breast tumor microbiome. Since joining our community here at UTHSC, Athena has demonstrated consistently high levels of productivity and received numerous awards and accolades. And that includes being selected as one of only 8 American Society of Human Genetics scholars for 2019 through 2021. And Athena was also recently awarded an R01 to study microRNA-based epigenetic approaches to induce fetal hemoglobin. She has authored 36 peer-reviewed publications, including 18 of those published within the last 5 years. And I also want to mention that in addition to this outstanding research career, Athena is also actively engaged in community outreach. And she has served as a volunteer tutor for minority students, an educator at the Sister Strut Breast Cancer Walk, and as a motivational speaker to encourage participation in genetic testing to better understand the genetics of breast cancer disparities in Black women. So Athena, welcome, and thank you for sharing your research with us today. And I just want to add one thing. If you could save your questions till the end of the talk, we should have 10 or 15 minutes. So take it away, Athena.
Thank you so much, Megan. That was a lovely, very lovely introduction, and it has motivated me now even more in this presentation. So thank you everybody for having me. I miss seeing your faces on campus. So first, I just want to say I have no financial interest to disclose. And here I wanted to first share that, so patients with sickle cell disease, they experience severe pain and increased risk of death. This is a painting by Hertz Nazier, who's a Haitian paint artist and author. And so he lives with sickle cell disease, and he utilizes his art to advocate and raise awareness for sickle cell disease and science education. So to illustrate his experiences with pain and suffering from sickle cell, he describes how he went blind for 2 weeks in his early 20s. And to this day, he still can't see out of his right eye due to a blood clot caused by sickle cell. He now creates enlarged reprints of photographs, one of which shows here him as a young boy in his hospital bed with his mother. And Hertz's painting here, titled Sickle Cell, Portrait of Pain, depicts his unbearable pain and suffering patients like himself have to endure. It's one of the most common inherited red blood cell disorders that affects over 20 million people worldwide, and over 300,000 infants are born with sickle cell anemia each year. So sickle cell anemia is more common among people of mostly African ancestry. Approximately 1 in every 365 African-American births are diagnosed with sickle cell disease. However, sickle cell also affects other racial groups, including those of Hispanic, Caribbean, Middle Eastern, Eastern Mediterranean, and Asian descent. Approximately 1 in 16,300 Hispanic American births are diagnosed with sickle cell disease, and about 3 per 1,000 white infants and 2 per 1,000 Asian, Native American, or other Pacific Islanders are born with the sickle cell trait. Now, there are different forms of sickle cell disease, and the majority of individuals with sickle cell disease have hemoglobin SS. But some make a different type of abnormal hemoglobin, like hemoglobin beta thalassemia, hemoglobin C, hemoglobin D, or hemoglobin E. And hemoglobin SS disease, also called sickle cell anemia, is the most severe type of sickle cell disease. In this type, the body only produces hemoglobin S molecules. So sickle cell anemia is inherited in an autosomal recessive manner. So the disease develops when an individual receives 2 copies of the mutation in the hemoglobin subunit beta, or also referred to as HBB gene, one from the mother and one from the father. So a person with just 1 copy of the mutated gene is said to have the sickle cell trait. Although they are a carrier of the disease, these people generally do not display any symptoms, but can pass the disease on to their children. So sickle cell anemia results from a mutation in the hemoglobin subunit beta gene, and hemoglobin, or HBB, contains the blueprint for cells to make the protein hemoglobin. As you can see here, portion, hemoglobin molecule bind to a missense mutation in the beta subunit. Okay, my internet says it's unstable, but you can still hear me. I see Megan's head shaking good. Of the beta globin locus, which is located on chromosome 11. So in individuals with sickle cell anemia, the codon GAG here, which is found in normal red blood cells, is replaced by GTG in sickle cell red blood cells. This results in the replacement of a hydrophilic amino acid, glutamic acid, with the hydrophobic amino acid valine at the 6th position. This substitution creates a hydrophobic spot on the outside of the protein that sticks to the hydrophobic region of an adjacent hemoglobin molecule's beta chain. This further causes clumping of hemoglobin S molecules or polymerization into rigid fibers, causing sickling of the entire red blood cells. So hemoglobin S polymerization is favored under low or deoxygenated conditions. So as the cells cycle throughout the body, like up to 4 times per minute, going from high to low oxygen environments, they keep polymerizing and unpolymerizing over and over, and that stresses and damages the red blood cells. This results in hemolytic anemia and a likelihood of blocked small blood vessels, which in turn can cause vaso-occlusion, as depicted here, or clogged blood vessels. And so vaso-occlusion typically causes acute complication, including ischemic damage to tissues, resulting in severe pain or even organ failure. So strategies to prevent this hemoglobin S polymerization under low or deoxygenated condition is warranted. So a natural way scientists are actually looking to prevent hemoglobin S polymerization is through induction of fetal hemoglobin. This is because fetal hemoglobin, which has those two gamma subunits and 2 alpha subunits instead of 2 beta subunits in adult hemoglobin, the fetal hemoglobin has an increased affinity for oxygen at all partial pressures compared to adult hemoglobin. Thus, a fetal can load and unload oxygen readily, and hemoglobin disorders won't become apparent until after birth, when the beta subunits start replacing the gamma subunits. So this is referred to as hemoglobin switching. So in most people, levels of fetal hemoglobin, represented by this blue line, it begins to decline in months preceding birth, while levels of adult hemoglobin, here in the represented by this red line, increase. It's also referred to as hemoglobin A. Since hemoglobin A contains the component of hemoglobin defective in patients with beta thalassemia or sickle cell disease, these patients do not begin to experience symptoms until several months, roughly about 3 to 6 months after birth. And so this is, which is after hemoglobin switching has occurred. So the expression of these globin genes is controlled by chromatin looping and epigenetic modifications, including DNA methylation. In adulthood, you have the upstream enhancer locus control region, and it's comprised of about 5 hypersensitive sites, and they're occupied by various DNA binding proteins, such as illustrated here, and they're brought into close proximity to the beta globin gene, as shown here. This interaction, it mediates long-range enhancer promoter interactions and beta globin gene expression. And beta globin gene activation is associated with an open chromatin state containing unmethylated CpG residues represented by these open circles, and it also has active chromatin marks such as histone 3 lysine 4 trimethylation and histone 3 lysine 27 acetylation. And by contrast, there's a silent epsilon and gamma globin genes that do not interact with this locus control region. They're displaced into a closed state, and that's characterized by the histone trimethylation and acetylation marks. So therapeutic interventions aimed at reactivating fetal hemoglobin is an effective strategy for ameliorating the disease severity of sickle cell disease. And reactivating gamma globin expression also improves long-term survival, since mechanistically, fetal hemoglobin blocks hemoglobin S polymerization and red blood cell or erythrocyte sickling. And so individuals who naturally express high levels of fetal hemoglobin beyond infancy thus receive some protection from sickle complications, including fewer vaso-occlusive crises, less renal damage, less pulmonary hypertension, fewer strokes, and longer survival. However, therapies aimed at inducing fetal hemoglobin are limited. Few drugs have been approved to increase gamma globin. The most widely used fetal hemoglobin inducing drug is hydroxyurea. And hydroxyurea, it's helped many patients with sickle cell disease. However, there are several side effects associated with its use, and many patients resist taking hydroxyurea due to its chemotherapeutic side effects. Another promising agent is decitabine, or 5-aza-2-deoxycytidine. It's a DNA methyltransferase inhibitor that de-represses or reverses epigenetic silence gamma globin expression. And decitabine has demonstrated robust induction of fetal hemoglobin and total hemoglobin levels in sickle cell patients treated intravenously. However, high concentrations of decitabine does produce off-target anti-metabolite effects as well as cytotoxicity, in significant part via its uridine moiety degradation products that can misincorporate into the DNA. And so ideally, treatments for sickle cell will reactivate gamma globin expression and induce fetal hemoglobin without causing bone marrow toxicity and avoid some of these off-target side effects. So an attractive class of small molecules under development of therapeutic intervention are microRNAs, or I like to call them MERS. So microRNA therapeutics, they've been investigated, or highly investigated actually, due to their capacity to restore control of aberrantly expressed genes that cause a wide array of human diseases using microRNA mimics or inhibition of microRNA function by anti-MERS. And several microRNAs have been investigated as potential fetal hemoglobin inducing agents by targeting regulators of gamma globin and beta globin expression. And in a study with Dr. Betty Pace at Augusta University, we showed that a particular microRNA, miR-144, it functions as a repressor of NRF2, which plays a role in globin gene silencing, and HbF, or fetal hemoglobin expression, in patients with sickle cell disease. And we use a genome-wide microarray-based microRNA screen of RNA isolated from reticulocytes. So what are microRNAs? MicroRNAs are small, single-stranded, non-coding RNA molecules containing about 20 to 28 nucleotides. They're conserved across species and function in RNA silencing and post-transcriptional regulation of gene expression. And so microRNAs, they're produced in a tightly regulated pathway. The biogenesis of microRNAs begins with their transcription by RNA polymerase II. And then these prime microRNAs are processed co-transcriptionally by microprocessor RNase DROSHA, along with the cofactor protein DGCR8, which then binds to the primary microRNA transcript. And then following this microprocessor cleavage, this exportin 5 transports the pre-microRNA hairpin into the cytoplasm from the nucleus, and there the pre-microRNA is cleaved by an RNase called dicer, resulting in this microRNA duplex. And then the microRNA duplex is further incorporated into an RNA-inducing silencing complex, and with the help of some transactivating response elements of RNA binding proteins, the mature microRNA is then loaded into an Argonaute subunit of the silencing complex. And then following some unwinding and strand selection, the mature microRNA is now capable of target recognition. And in binding of the mature microRNA to this RNA-induced signaling complex, it leads to targeting of messenger RNAs with complementary sites, and it results in translational repression or mRNA degradation. And so one particular microRNA of interest that I've been studying is MicroRNA29B, due to its ability to target the 3' untranslated region of the de novo DNA methyltransferases DNMT3A and DNMT3B, resulting in its gene silencing. And in my previous work, we demonstrated that MicroRNA29B, which is mechanistically similar to decitabine, decreased expression of DNMTs in breast cancer cells when introduced in cell culture in breast cancer cell lines. And this introduction restored control of aberrantly expressed tumor suppressor genes involved in cell cycle control that were initially turned off due to promoter hypermethylation. So we hypothesized that maybe MicroRNA29B may function similarly as decitabine, which also functions as a DNA methyltrace inhibitor and treatment for sickle cell disease. So here, interestingly, using the same genome-wide microRNA screen that we did in the paper that I showed you previously for microRNA144, we found that MicroRNA29B, the mRNA expression levels were significantly increased in reticulocytes of sickle cell patients with high levels of fetal hemoglobin compared to those with low levels of fetal hemoglobin. And so these were patients of Dr. Betty Pace, who's also my collaborator and on my R01 grant. After she obtained informed consent, the blood samples, they were collected from children with sickle cell disease, a total of 12 of those children. They weren't taking hydroxyurea therapy because hydroxyurea induces fetal hemoglobin. So we want to test our microRNA. So we don't want them on any fetal hemoglobin inducing agent. And the children range from age 4 to 20 years of age. And so male and female participants were equally distributed in the high and low fetal hemoglobin groups. And so here the clinical phenotype data included fetal hemoglobin levels, complete blood counts or CBCs with differential and reticulocyte counts. But what I want to just point out here are the p-values. There was no significant difference in hematological parameters between the high versus low groups. And the individuals with low levels of fetal hemoglobin, the average fetal hemoglobin was about 3.4%, and high range, the average high fetal hemoglobin levels, or the mean, was 23.4%. And these extremes were used for further microRNA analysis. And so our initial findings, these initial findings further supported our investigation to the functional role of miR29B in gamma globin gene regulation. So here Our initial preliminary findings were done in KU812 leukemia cells. And here we just show that introduction of increasing concentrations of the miR29B mimic into KU812 leukemia cells significantly increase gamma to beta globin mRNA expression levels, or their ratio, compared to control. So here we have 25 nanomolars of miR29B, 50 and 100, and there's an increase in that ratio in a concentration dependent manner. Similarly, the percentage of F cells increased in a concentration dependent manner, or the percentage of HbF or fetal hemoglobin positive cells were significantly increased in a concentration dependent manner following miR29B treatment. And we also observed this at the protein level, and we did get a slight reduction in adult hemoglobin A protein levels following miR29B treatment. So based on our preliminary findings, we sought to determine whether miR29B mediates gamma globin activation and fetal hemoglobin induction in normal and sickle erythroid progenitors. So we've established a collaboration with Dr. Adams-Graves, Dr. Ugochi Ogu, and Dr. Kenataga on campus in the Regional 1 Diggs Cross Comprehensive Sickle Cell Clinic to obtain blood samples from sickle cell patients for these studies. So here are just some inclusion and exclusion criteria for the patients that we selected for our studies. So all patients with a known diagnosis of sickle cell disease, either hemoglobin SS or beta thalassemia, are currently being approached for enrollment. And the diagnosis of sickle cell disease is confirmed by HPLC analysis. And patients on a stable dose of hydroxyurea were excluded, are not eligible, since we're testing the potential fetal hemoglobin inducing agent, our MicroRNA29B. Also, as patients receiving chronic transfusion therapy are excluded, since chronic transfusion therapy may alter blood work, as well as patients with acute fever, vaso-occlusive crises, splenic sequestration, or acute chest syndrome within the past month, since it also may alter their blood work, secondary to their acute sickle cell complication. And so also patients not providing informed consent were excluded. And thus far, we have consented and obtained a 10 ml blood sample from 26 adult sickle cell patients. So once we receive the blood samples, we process the blood samples with FICO to isolate peripheral blood mononuclear cells. And so this is just a depiction of what we do. So we have our blood samples, and we add this component called FICO histopake to isolate these peripheral blood mononuclear cells in this little white section here, this little line of white, which contains our peripheral blood mononuclear cells. It's kind of hard to see, but it's a little line of white, well, little white line that contains those cells. And so these peripheral blood mononuclear cells were further cultured in conditioned media using a two-phase erythropoiesis culture system to generate our erythroid progenitor cells, or premature red blood cells. And this is just a flow chart of that process. And so basically, sickle erythroid progenitors, or adult normal CD34 stem cells, they were introduced on day 8 with human MicroRNA29B, or our scramble control microRNA, or scramble control mimic. And then after 48 hours, we measured expression of genes involved in DNA methylation, gamma globin gene expression, and measurement of the percentage of HbF, or fetal hemoglobin positive cells. So our results in normal adult erythroid progenitors showed that the ratio, again, of gamma to beta globin was significantly increased after treatment with miR29B alone, which you can see here for 50, and we use 50 and 100 nanomolars here. And it was higher, so we had a higher gamma to get beta globin expression levels for these miR29Bs, which was slightly higher than decitabine treatment alone. And we also determined the percentage of F cells, or HbF positive cells, in in erythroid progenitors stained with FIDC anti-fetal hemoglobin antibody under the different treatment conditions shown here. And so here, just to point out, the percentage of HbF positive cells or fetal hemoglobin positive cells also significantly increased with treatment with miR-29B alone and when combined with decitabine, although it was not an additive effect. And so given that decitabine and miR-29B combined didn't produce an additive effect, we also performed studies with hydroxyurea. And so primary erythroid progenitors, they were treated with 75 micromolar hydroxyurea or the miR-29B mimic at 50 or 100 nanomolars alone or in combination with hydroxyurea. And so treatment with 100 nanomolar MicroRNA29B, it increased the percentage of HbF positive cells or fetal hemoglobin positive cells to 8% alone. But with hydroxyurea, the percentage of HbF positive cells. But combined treatment resulted in a slightly increased percentage of HbF positive cells as compared to treatment alone. So in our next set of experiments, we sought to determine whether miR-29B had an effect on expression of genes involved in DNA methylation and gamma globin promoter methylation status, since miR-29B acts as a DNA methyltransferase inhibitor, similar to decitabine. So here we observed lower DNMT3A as well as lower DNMT3B mRNA levels. Excuse me, give me 1 second, I need some water. So we observed lower DNMT3 mRNA levels in cells transfected with miR-29B compared to control scramble control, which did not change for combined treatment with decitabine. And at the protein levels, decreased expression significantly MT3A and DNMT3B and the gamma globin repressor protein MIB, but it significantly increased fetal hemoglobin levels. And we further evaluated levels of gamma globin promoter methylation status by measuring levels of 5-methylcytosine, which is a marker of DNA methylation. We also measured levels of unmodified cytosine residues at the -54 CpG site in the 5' cap of the gamma globin gene using an epimark methylation assay and real-time PCR primers, as previously described in this paper that we use the primers from. And so basically what this shows is that we observed that miR-29B decreased gamma globin promoter methylation as indicated by a significant decrease of 85% of 5-methylcytosine levels when compared to the untreated control or scramble control erythroid progenitors. And these findings further support the ability of miR-29B to mediate demethylation. And so we also performed the same studies. So initiation of sickle erythroid progenitors at day 7, 12, and 14. This is from an individual sickle cell patient. What we did, we further confirmed that miR-29B indeed increased levels of fetal hemoglobin, levels in erythroid progenitors, and these are patients not a patient not on hydroxyurea. So you can see at a 100 nanomolar miR-29B increased the percentage of fetal hemoglobin positive cells at 63% as compared to scramble control cells. And here the scramble control is high. So we're testing these experiments using a different scramble control to understand why that this is high. But overall, you can see that there is a significant increase in the percentage of HbF positive cells as compared to untreated and scramble control. And so these findings, it further supports or demonstrates that miR29B induces fetal hemoglobin in sickle erythroid progenitors and supports a potential for clinical development. And so our findings support a role of miR29B in fetal hemoglobin induction by targeting DNMT3 enzymes, decreasing gamma globin promoter methylation levels, and downregulating MIB expression. And we have further published these findings in the British Journal of Hematology. And I think the students have had the opportunity to read this, but here it is made available and for you to further assess it. Okay, so basically these studies are recruitment efforts. They're going to support the first aim or in hemoglobin general and sickle erythroid comparison. So how long does fetal hemoglobin levels remain elevated? And the short answer to that is that we don't know yet. However, we're testing the efficacy of miR29B in a preclinical sickle cell mouse model in sub-aim B of our R1 grant. And so for this sub-aim, I have a collaboration with Dr. Betty Pace at Augusta University, who maintains a town sickle cell mouse colony. And she is testing the MicroRNA29B in fetal hemoglobin induction in this particular mouse model. So these mice carry human alpha globin, gamma globin, and beta S globin genes and exhibit phenotypes including chronic hemolysis and sickling similar to human sickle cell disease. So basically, to establish the optimal dose of miR29B mimic that induces levels of fetal hemoglobin, treatments will be delivered by continuous infusion over 28 days using surgically implanted mini osmotic pumps. And treatments will consist of 4 groups, 4 mice per group. It'll consist of 4 mice per group, excuse me. Consist of 2 males and 2 females for miR29B or scramble control mimic at 2, 3, and 4 milligrams per kilogram per day. And we'll also have a group of water, which will serve as the vehicle control, or hydroxyurea, and the mice will receive 100 milligrams per kilogram per day. And so we'll also do tail vein blood sample. Blood samples will be collected in
Oops, we lost that last bit.
I'm so sorry. Oh my.
Oh, that's okay. That's technology. The Zoom.
Can you hear me now?
Yeah, I can hear you, but it's a little spotty. Maybe just try, because we're almost, yeah.
Okay.
We've almost made it.
I'm sorry. Oh gosh. All right.
So I think
So
we may have missed the last part of the description of the experimental study. You were talking about the tail vein injections.
Okay. Why won't my computer allow me to, okay. Oh, here we are. Okay. So this part, so basically we established optimal dose of miR-29B mimic. So we'll deliver continuous infusion over 28 days using surgically implanted mini osmotic pumps. And treatments will consist of the 4 groups, 4 mice per group, consisting of miR-29B or the scramble control mimic water vehicle or vehicle control or hydroxyurea. And this will be over 28 days. And tail vein injections will also be a separate study to compare how treatment with mini osmotic pump, where you have continuous delivery, how effective that is versus the tail vein injection. And so basically, blood samples will be collected at 0, 2, and 4 weeks of treatment. These mice and tissues will be harvested, including brain, lung, liver, and so here are preliminary results. Okay, our preliminary results in Towns mice aged 3 to 4 months old, consisting of 3 mice per group. Here we use, she used 2 males and 1 female. Basically here, we just show that treatment with miR-29B had minimal effects on hemoglobin levels or white blood cell counts differential. And it's shown here, we observed a significant decrease in total white blood cell counts, as well as increase in platelet count at weeks 2 and 4. Of interest, the reticulocyte counts decreased for miR-29B, consistent with less turnover in the blood marrow since neutropenia didn't occur. And here, during the treatment period, there was also no adverse side effects in behavior, and all mice had steady weight gain, as shown here at 2 and 4 weeks. At the end of treatment, also pumps that were removed, they were empty, which confirmed that all the MicroRNA29B medication was delivered effectively. And real-time PCR analysis of peripheral blood RNA further confirmed a highly significant nine-fold increase in miR-29B at 2 weeks, after 2 weeks of exposure, which decreased to 4-fold by week 4 compared to scramble control here. And there was no significant change in miR-29B levels for water or hydroxyurea for all those weeks. And lastly, well, almost lastly, so to determine the ability of miR-29B to induce fetal hemoglobin at the protein level in vivo, we performed Western blot analysis of red blood cell hemolysates. And at the protein level, we observed a 4.2-fold increase in fetal hemoglobin levels at weeks 2 and a 2.1-fold increase in fetal hemoglobin level at week 4 after miR-29B infusion compared to scramble control. Now, lastly, to substantiate a phenotypic effect of miR-29B, we performed a sickling assay in mice using freshly collected blood incubated under 1% hypoxia or low oxygen conditions for 6 hours. And so here we observed a 33% decrease in the percent of sickle cells at weeks 2, which was maintained at 20% inhibition at week 4 compared to scramble control, which increased. So finally, in conclusion, our findings support a role of miR-29B in fetal hemoglobin induction by targeting DNA methylation machinery. And so these studies are ongoing, and we hope to establish new collaborations with other investigators and clinicians with expertise in sickle cell disease, globin gene regulation, and drug delivery approaches. I would like to acknowledge everyone on this slide, particularly the sickle cell patients treated at the Dix Cross Clinic. My lab manager and assistant, Alana Smith, Miss Alana Smith, she works really hard and generated most of this data shown in the presentation. My new postdoc, Dr. Gu, and Dr. Adams Graves, Dr. Ataga and Dr. Ogu, who are the clinicians that help us to get blood samples from sickle cell patients. I also like to thank Dr. Betty Pace at Augusta University, who serves as my mentor as well as co-investigator on this R01 grant and her lab members. I would also like to thank Dr. Mitch Weiss at St. Jude Children's Hospital and Dr. Yao, who helped us get the erythropoiesis culturing system off the ground and running in our lab. And last but certainly not least, I would like to thank funding. The Cornett Award from the University of Tennessee. UTHSC was very instrumental in providing the preliminary data that we needed to generate the R01 data, or to get the R01 grant actually. CITG, thankful for those funds and thankful for newly R01 grant. And I welcome any questions that you have, and sorry about the complications with the internet connection.
Very cool talk, Athena.
Thank you, Rob. Sorry about that. This has never happened to me in the middle of a talk.
It was no problem, really.
Gosh.
Athena, that was a beautiful presentation.
Oh, thank you. Thank you.
I have just a couple of minor questions. One is, how do you design the MIR to be protected from nucleases?
How do I, well, okay, that's a good question. So this primarily, I guess, refers to the in vivo studies.
Correct.
Yeah, so we purchased them from Dharmacon, and they basically covered them in a cholesterol. I forget exactly how they actually developed the mimic, but they developed it to where it has like a cholesterol moiety on it to protect it from degradation. But that's one of a concern about degradation by RNases and things. So we're really trying to develop collaborations with people that have, drug delivery approaches and things like that. So that's always the question that I get. So yeah, maybe something like how the Moderna vaccine, how it's being delivered, we can kind of mimic that.
Yeah, I think that's a good possibility. The other question was related to your scramble effect on HbF. Do you think that's potentially a clue to what is an off-target effect that may be useful? In other words, and as part of that question, do you think, or do you have any data currently indicating whether that scramble is affecting methylation, or is it an independent effect?
So that's a great question. We don't know the answer to that yet, but we're going to do some RNA-seq to determine whether or not what other off-target effects, what other genes might be upregulated or downregulated when using this mimic. But we're also going to, just to make sure that it's not a technical issue, we also will purchase another, control mimic. But yes, we're definitely going to do global methylation analysis as well as RNA-seq just to see if there's any off-target effects.
So it might be very useful as another adjunct, right?
Yeah, exactly. Yes, definitely.
I wonder if you combine the scramble with your MIR, whether you actually get an additive effect.
Oh, that's a really good point. I didn't even think about that. That's something that we can definitely check and just see what, I would expect it to have like an inhibitory effect, but yeah, that's a good point.
Thanks a lot.
Thank you.
Lovely presentation, Dr. Davenport. I have one question. So you spoke about the mouse model collaboration. So my first question is, in mouse model, what was the rationale to choose like 3 to 4 months old mice, male and female?
So yes, that's a really good question. So we chose, Dr. Pace, she chose that particular age group because possibly at that, so I'm not sure if at that age group, that's where the levels of gamma globin to beta globin, that switching kind of occurs before then. So it occurs slightly before there, but it could be, that's a pretty significant age to when you can see, where you have your most significant change in gamma, that hemoglobin switching, basically.
So was the mice, this group of mice, were they wild type mice and they fed microRNA, or like it was hemoglobin B mutated mice?
Yes, they have the mutation in the hemoglobin S. So their sickle cell, that's how they actually demonstrate a phenotype similar to human sickle cell.
Okay, so in that case, just two males and two females, if I understood correctly, just they chose two males and two females. Are they enough? At least they should have n is equal to 3 to 4 to see the effect and to confirm the effect.
Yes, so we'll have multiple groups. So for the preliminary data that I showed today, it was just two females or one male or vice versa, just for generating some preliminary data of the MicroRNA29B in the mice. But yes, that's a great point. We're definitely going to increase, there'll be multiple replicates, different groups of mice to test the microRNA.
Yeah. Thank you. Thank you very
You're
much.
welcome. Thank you. Thank you for your question.
Hey, Dr. Davenport.
Hello.
I have a question kind of near the end of your presentation, and you'll have to pardon me. You were cutting up half the time, so I may have missed something that you talked about earlier. But you brought up hypoxia, and I wanted to know exactly within what context that was and whether or not this could result in a confounding kind of circumstance underneath like the Bohr effect.
So when I say hypoxia, so she exposed the mice under, 1% hypoxia or low oxygen conditions, or it was 21% atmospheric oxygen conditions for 6 hours. So at the low oxygen levels, generally when you put the blood of sickle cell patients under the low oxygen conditions, such as these, that will cause the cells to sickle or cause these polymerization to form because there's no oxygen being able in the hemoglobin molecule in that beta subunit. So that causes the hemoglobin S molecules to kind of sickle or become packed together.
And this would be different from relative acidosis that may, like, was there a way to actually control for that, or at least look into to see if there was any differences within that pH and oxygen tension level?
So seeing if there's a difference in the pH or oxygen, I guess I don't understand your question.
Yeah, so just like in like the Bohr modeling system, you see like the overall decrease of oxygen typically associated with decrease in the pH variables. So if you were solely focused on the oxygen or lack thereof, and you see a result in your cells, is it actually fully associated with that, or is it just a confounding event occurring within the pH and the oxygen levels alone?
No, I mean, it's definitely due to, I think, to the pH and the oxygen levels alone. I mean, it's not a confounding effect. It's just something that just naturally happens under these low oxygen conditions. Does that make sense, Winter?
Yes, I
don't know if I answered your question.
Thank you.
No, great question. It makes me, yeah, I have to think hard on that one.
I actually had a couple of naive questions. So you had done the microRNA screen and then tested MIR29B it was, but there's other family members of MIR29, and I guess they have like the same seed region. So it's fascinating to me. I mean, do they all target the same DMNTs, or is it just that one? And it's fascinating if it's having the same seed region, they have that unique targeting.
Yeah, so that's a good question. So we looked at MIR29A as well as MIR29B and MIR29C. And when we did the microRNA screen, that there was no significant difference in expression levels for those particular microRNAs. And we did do some preliminary data in those KUH12 leukemia cells just to see what effect it had on expression. And we didn't see, I want to say, yes, we didn't see any significant increase in gamma globin. So that's why we focus more on MIR29B. And my background in looking at that particular microRNA really kind of furthered the study to kind of investigate that specific MIR29B.
And so that MIR29B, that's the only one of the family members that has the effect on the DNA methyltransferases. Oh, it's really interesting.
Yeah. And I think it has more of a binding affinity, or I don't know if that's the right word, with the DNMT3 enzymes compared to the others.
That's cool. That's interesting. Then I had another question I was trying to understand. So, because I think at some point you had shown that so surprising to me that some patients still do have like high fetal hemoglobin, even in adulthood. And I think you said they were like 23.4% is the mean. And some of them maybe even had 30.
Yeah, so that was, and these were just extremes. So, but yes, those were extreme numbers. But some patients, it's called HP. Oh gosh, how could I forget the name? It's basically where some individuals are naturally protected from, or not naturally protected, but they have higher levels of fetal hemoglobin, and because of that, they have less severity of complications and clinical outcomes.
So I guess my question was, how much do you have to increase that fetal hemoglobin? And are you seeing that like enough of that boost when you have the MIR29?
So that's a good question. So different studies have shown different percentages of fetal hemoglobin. What I've seen is roughly, if you maintain at least 20% levels of fetal hemoglobin, if you maintain at least 20% or higher, then a lot of the side effects are gone. So in our study, it's preliminary, and I would hate to say, of course, I want it to work, definitely. But if we can maintain those levels, that would be great with this microRNA. That would be fantastic. And I can imagine maybe how we get the COVID vaccine, the Moderna, maybe a once a year. It would probably be more often where patients would have to get injection with the microRNA, but that's a long ways from here.
Yeah. All right, great. Thank you. That was very informative.
Thank you.
Any more questions, graduate students?
I had a question in the chat, Athena.
Oh, is the Townes mouse human allele semi-dominant? Do the heterozygotes have any abnormalities? Human allele. Is it semi-dominant? That's a great question. So if the allele is so if the sickle cell, it's a recessive disorder. That's a good point. I need to check that out.
Yeah, obviously sickle cell itself has a very important semi-dominant function, right? In terms of relative resistance to malaria.
Yes.
I was wondering whether you could map modifiers by crossing the Townes mouse with the BXDs, but you'd have to have something interesting to evaluate in the heterozygotes.
Right. And You know, I've mentioned this to Dr. Pace before, and I want to say that she's, oh gosh, it's been a while. Whether or not these mice, they can't because they don't have. It was, I don't want to say wrong, but if the heterozygotes have any abnormalities.
Yeah, one of your slides made the case that you're interested in collaborations. That's one that would interest me. So
Okay,
if-
perfect.
if there is
Yeah.
any kind of semi-dominant action, let me know.
Okay, I definitely will.
Okay.
Any questions from the graduate students?
Yeah, they probably didn't get everything because I was cutting in and out. Ah.
Well, I guess if there's no more questions, or if you have any additional last comments.
Yeah, if anybody has questions or anything, just reach out to me. I'll be more than happy to meet with you to discuss it for sure.
All right, well, thank you. That was a really exciting, fun, and interesting talk. And
Thank
thank all of
you.
you for joining.
Thank you. Have a great weekend.
Bye, everybody.
Bye-bye.